Hello, everybody. Welcome to our last official chapter. This is chapter 9. This chapter is mostly on graphing. So, graphing is going to be a super important part of this lab because, well, one, it's science, but also because for your oral report and your long-term experiment, you will have to graph. So, we're going to talk a good bit about that and how to graph your scientific data. First thing I want you guys to know and note in your book, anywhere you need to, you can start on page 75 of your lab manual, probably the best place for it. But you need to know that your t, so your t value, cannot be negative. So your t value will never be negative. The website that we provide for you guys does often give you a negative t value. Whenever this happens, just take it and take the negative off, but use the same number. So just make it positive. But if you do have a negative t value, I will have to count off points. So make sure you pay close attention to that. So we're going to get into graphing a little bit. So according to your lab manual, the first step in constructing a scientific graph is to decide which type of graph is appropriate. So there are many different types of graphs to suit various types of data. You already learned the difference between qualitative and quantitative data. If you need a reminder, go back to chapter 1. Data also can be expressed as continuous or discontinuous. To select the best type of graph to fit your experiment, you must consider both the dependent variable, where the results are collected, and independent variable to determine whether they are continuous or discontinuous. So continuous data are quantitative values collected by measurements. So some examples of this include length, time, temperature, and speed. When data changes, it continues from one value to another. For instance, when time passes, it continues from 1 second to the next with all the fractions of a second in between. And as temperature rises, it continues from 1 degree to the next with fractions of a degree in between. Continuous data can always be more precise by adding decimal points or fractions of the unit measure. Discontinuous data can be quantitative or qualitative. It is gathered by counting or categorizing. The categories are discrete and do not continue from one to the next. Some qualitative examples are males to females, blood type, number rolled on dice, and number of cockroaches. If you roll a die, the dots on the top surface fall into the category of 1, 2, 3, 4, 5, or 6. The value cannot be more precise because there's nothing in between. A roll of 4 does not increase up to 5 or decrease down to 3. Similarly, if you census cockroaches, it is a whole number. You cannot count a half of a cockroach. We are now going to look at the different types of graphs. Line graphs are typically used to relate continuous data results to an experimental variable of continuous data. A bar graph is used when you have continuous data for one variable and discontinuous data for the other. And finally, a pie chart is used if both variables are discontinuous. Here are some examples of each. So we are now going to go through various graphing rules. So first of all, the independent variable should be your horizontal axis. Your dependent variable should be your vertical axis. You need to always make sure you label your axes. This is very important. Label the unit of measurement. So if you are doing how long your MHC takes to complete a maze, your variable needs to be time, and in parentheses, you need to write seconds. You have to have a title that explains your graph. Your title should not be 1 or 2 words, and it should also not be a paragraph. Make sure your graph title has enough explanation that I can glance at it and know exactly what I'm supposed to be looking at. Include a legend if necessary. It's not always necessary, but sometimes it is, so make sure it is included if it is necessary. Your graph also needs to be systematic yet simple. This essentially means that I should be able to read it and I should be able to look at it and get all the information I need about your data. However, it should also not be so complicated that it takes me an hour and a half to read, okay? Pie charts also do not have axes. So if you're using a pie chart, then you don't have an axis at all. So here is an example of a line graph. So line graphs are continuous versus continuous data. In this particular example, say your pet gerbil just had a litter of babies and you wanted to track their growth, a line graph would be an excellent choice. You could weigh them from the time they're born and take a mean of all the baby's weight for the next 10 days. The result, or dependent variable, is their average weight. On what does the weight depend? Their age. So, from gram to gram, as age changes, it continues from day to day. Weight and age can both be measured more precisely by adding a decimal point to grams and hours to days. A line graph of this experiment allows you to visualize the steady growth of the newborns with only slight variation. So, as you can see, this graph has a title. It has a label on the x-axis or the horizontal axis, as well as the y or the vertical axis. So your dependent variable is going to be your vertical axis. Your independent variable is going to be your horizontal axis. Units of measurement are both present. You have grams and you have days. You also have a very descriptive title. And it is systematic and simple. There's no legend because there's not one needed for this particular graph. The next graph we're going to look at is a bar graph. So a bar graph, the data is going to be continuous versus discontinuous. So in this case, we are looking at an example where you tested different brands of fertilizer on tomato plant growth. The independent variable, brand of fertilizer, is discontinuous. The dependent variable, plant height, is continuous. The height depends on the type of fertilizer used. Although the fertilizer type is categoric and cannot be made more precise, the height of the tomato plants can be more precise than a measurement of whole inches. The long-term and short-term cockroach experiments in this lab will almost always require a bar graph. So, in this one, you can see, again, you have your dependent variable on the vertical axis. You have your independent variable on the horizontal axis. You have your units of measurement for both. Fertilizer does not require a unit of measurement for this one because it is discontinuous. However, your continuous, which is going to be your height, is required, and you have inches. You also have a descriptive title for this one. You have systematic and simple, and again, there is no legend because there's not one needed. The last graph example we're going to look at is the pie chart. So remember, pie chart is going to be discontinuous versus discontinuous data. So in this case, we are going to look at blood types. So pie charts are particularly handy in depicting percentages or proportions of a whole. So for example, in this particular example we're looking at, if you investigated the ABO blood type of all students in your biology class, you could represent your data in a pie chart. Blood type is the independent variable, and the distribution is the dependent variable because the proportion of students depends on the blood type. The blood type is categoric discontinuous data because everyone falls into one of the 4 categories. The distribution is also discontinuous because each student is fully one blood type. Although percentages can have decimals, the percentages are based on students and there are no fractions of a student. So, since it's a pie chart, you do not have an axis. Your dependent variable is going to be your percentage. Your independent variable is going to be each different section. So you have A, B, AB, and O blood. There are no units of measurement for this one. Has a descriptive title. And this one does have a legend. This legend is very clear. You can clearly tell which is the different blood type. For this one, make sure when you give this, your colors are different enough that you can tell the difference. If you end up with a lot, then a lot of different sections or categories, make sure that it is easily readable. And again, this is systematic and simple to read. So now that we have talked through the different types of graphs that you're going to look at, here are 3 examples of graphs. So at the very top, you have a pie chart. All of these are on the same thing. So comparison of the weight of Madagascar hissing cockroaches according to sex. So take a second to look at these 3 different graphs. Which one do you think best represents the data that you are looking at? I'll give you a few seconds. You can pause the video if you want. So take a minute. Okay, so if you looked at this, you probably were able to figure out that we have continuous and discontinuous data. So this means that our line graph is out as well as our bar graph. So neither of these are going to be the correct type of graph we need. So we're going to end up needing a bar graph, or the one in the bottom right. So in this graph, you have your weight in grams. So you have your unit of measurement, and then you have on the horizontal axis, you have sex. So it is separated in male and female, and the student has taken the average of all of the weights that they took. So we're now going to look at some different examples. Okay, so we have now narrowed down our graph to bar graphs, right? So we're looking at the same example. However, these 2 graphs are a little bit different. So I want you to take a minute or 2 to pause the video and figure out what is wrong with either of these graphs. Are either of them correct? Are neither of them correct? So pause the video, let me know. Okay, so looking at the very first graph. This graph looks pretty good, right, from just a glance. So the title is great. It's the same title as we used in the previous example as well. You can clearly see the data. The vertical axis is labeled appropriately and has the measurements. However, this one is missing a horizontal axis. So in this case, just because you have the 2 different categories, which are male and female, does not mean that that is the axis title. So you still need an axis title. The best one for this is going to be typically sex. There might be 1 or 2 other ways that you could formulate that, but sex is probably going to be your easiest since you are, that's what you're comparing. For the second graph, this one is fairly similar as well. So this one does have both axes titles, as you can see. However, your vertical axis does not have your measurement. So it has weight, but it doesn't say how the weight is being measured. So this one needs to add grams to it as well. So neither were perfect, both needed a little work. So this next example, so you have 2 different graphs, both are still bar graphs. So I want you to take a second to look at these 2 graphs and figure out if there are any differences. Are either of them correct? Are neither of them correct? What needs to be fixed? Okay, so looking at the very first graph, for the most part, this graph is pretty okay, right? Looking at it, it has both of its axes titles. It's clear and easy to read. However, it does not have a concise title. So this title is only a couple words, and it really does no good. So you can, it says cockroach weights. Well, what does that mean? What type of cockroach? We have no idea that it's being compared. We have no idea that it's being compared by sex specifically. So a appropriate title is very, very important, especially in science. Now, looking at the second graph, this graph has a great title. And while its axes have titles, they're backwards. So this is something you have to pay very, very close attention to, because in Excel or many other types of ways to format graphs, it will let you do whatever you want. It will not tell you, oh, you did this incorrectly. So you have to pay very close attention to how you are doing this. So your axes need to be switched. So weight needs to be the vertical axis and sex has to be the horizontal axis in this case. So if your graph ends up looking like this, you did something wrong and you should turn around and redo it, okay? Just so you guys know. So the next example we're going to look at is fairly similar to the same. So with this one, the change is actually going to be very subtle. So I'll give you a second to look. Is there anything that you guys can find? So take a second, pause the video. Okay, so if you look at the first graph, it looks like male versus female is a very, very large difference, right? Well, if you go over to your vertical axis where weight is, you can see why. This graph starts at 40 and ends at 48. This is not a great way to do this. So your graph should always start at 0. If you don't start at 0, you end up with misconceptions like this. So with this one, it looks like there's a massive difference between male and female. However, it looks like the difference is only about 5 grams. So this isn't a very big difference at all. If you look at the second graph, so this graph is actually a really great graph. It starts at 0. You can tell that there is a difference between males and females. However, it is fairly small, which is exactly what we were seeing in this. So some other examples of things that I've seen often with students. So I have 3 different graphs up here. So I'll give you a second to look at all 3 graphs and figure out which one is the best. So one of these graphs is 100% correct and you would get full points on. So figure out which one that is. Pause the video, take a second. Okay, so if we're looking at this top graph, so this one isn't terrible. However, it would not get full points. It has a great title. Both of the axes are labeled clearly. Starts at 0. The data is going to be the issue. So in this case, this student took every single trial, and what it looks like they're doing is they're comparing each individual trial within male and within female. So this is incorrect. What needs to be done is all of the weights of the males need to be averaged together, and then all of the weights of the females need to be averaged together. So you should only have 2 bars total, male versus female. When you do this, you also do not need a legend at the bottom. So looking at the graph underneath that one, a fairly similar concept here. So in this case, they are essentially comparing different trials to each other. So what this means is if you had so say your experiment was to see how long it took a male versus a female to go through a maze. So in this case, what they're doing is they took a female and they took a male and they did trial 1. And in that trial, they're now comparing those 2 cockroaches. Well, what does that have to do with anything? So if you think about it, you took multiple cockroaches in this class to compare against each other. Just because you happen to pick these 2 specific ones on the first day versus say 2 other ones on the 4th day, does not mean that they need to be compared. There's no reason for them to be compared specifically. So again, with this one, you're going to take the average of all the males and you're going to take the average of all the females, and that's what you're going to compare. So knowing that, we can now figure out that this first graph on the bottom is going to be your perfect graph. So it has 2 bars, and that is it. It's comparing the average weight of males versus the average weight of females. All of the axes are labeled correctly, and it has a very concise title. All right, so the last thing we're going to look at is the homework for this. So this homework is super helpful for you guys because it gives you a chance to, 1, practice your graphs, which is something you will probably have to do forever in life. Learning how to read a graph as well as make one is important in most careers, not just science. But this graph will also be used in your oral report. So by going ahead and working on this, you will be able to have a little bit of a leg up for your oral report. So the first thing you're going to do, this is an individual assignment. So this means that you should not be working with your partners. I know that your partners have the same data. However, your graph should not look exactly alike. If you do, and I suspect that you are stealing other people's graphs, there will be points taken off for that. So please don't do that. I want you guys to get full points. The next is, this needs to be computer generated. So please do not write a graph on graph paper or anything else. It has to be computer generated, okay? Especially since you're turning it in on Blackboard anyway. So make sure it's computer generated. If you have questions or if you need help, go to YouTube. YouTube has a lot of great videos on how to do this. You should be able to figure it out pretty quickly. Make sure you have the correct type of graph. So this is greatly going to depend on the type of data you have, but make sure you use the correct graph. If you need more help, go back through this video, or you can also look at chapter 9 in your book. It starts on page 75, and it will be super helpful for you guys to figure out which type of graph you need to use. Make sure you have all the proper components of a good graph. So we went through all of those in this video, so make sure you have all of those. I will take off points for each one that you don't have. So this one is, you can ignore this one. So you don't need to do this because you're turning it in onto Blackboard. You're turning this in on the 15th of November. This is Sunday. It needs to be turned in by 11 p.m. If you have questions, please email me earlier in the day because I will not be awake by 11, okay? There are some extra directions on Blackboard for you guys under the submission, so please make sure you look at this. You're also going to add with this graph a hypothesis, so all of this should be focused on your long-term experiment. Make sure you have your data, your means, and you want to have your degrees of freedom, your t-value, and your p-value. So you're going to go ahead and run a t-test. This does not mean that you take a picture of all of the math portions that it gives you and screenshot it and put it on there. You need to know the specific difference between your degrees of freedom, your t-value, and your p-value, okay? Again, if you have questions, please let me know.